Hello, in this video we'll see phospholipids structure, function and synthesis. Phospholipids are backbone of cells. They create the cell membrane of all the 30 trillion cells of our body. They play a very important role in keeping things in the cells and also not letting things come into the cells from outside or leak out of the cell as well. We know that fats and lipids are hydrophobic because carbons are attached to more hydrogens so that they hate water molecule. But in case of carbohydrates, which has carbons attached to more oxygen molecules so that they can mix well with the water molecules. In this video, we'll see the crucial role of phospholipids in cell membranes and their structural contributions to the formation of the lipid bilayer, to their involvement in vital signaling pathways. That is, the diverse functions of these molecules are explained well in this video. So this is the structure of phospholipids. Phospholipids are a class of lipids, amphipathic molecules composed of hydrophilic head group, that is glycerol molecule, and two hydrophobic fatty acid tails. So this is the glycerol molecule, which is the backbone of phospholipid, which is consisting of the first two carbons are attached to two fatty acids, and the third carbon consists of phosphate group, which is surrounded by oxygen molecules. The hydrophilic head group consists of various combinations of functional groups, such as choline, ethanolamine, serine, or inositol, linked to the phosphate moiety. The fatty acid tails can be saturated or unsaturated, and their composition determines the physical properties of the phospholipids. Functions of phospholipids. Phospholipids' structural role in membranes. So this is a phospholipid. The phosphate group pops itself away from the rest and creates phosphate head, and then those two fatty acids form two fatty acid tails. So that means we have a part of phospholipid that loves water molecule, and the other part hates water molecule. These are nothing but the two fatty acid tails. So these molecules are spontaneously arrange themselves in a way in which the phosphate head is exposed to the liquid environment and the fatty acid tails actually embedded deep within each other, and they are not exposed to the liquid environment. So in that way phospholipids creates a bilayer with the phosphate head outside and fatty acid tails inside. So with this the phospholipids bilayer, they act as a selective barrier that regulates the entry and exit of substances in and out of the cells. The selective permeability allows cells to maintain internal conditions, that is, they control the transport of essential molecules and prevents the entry of harmful substances. Thus, phospholipids contribute to the fluidity and flexibility of the membranes, allowing for membrane dynamics and cell movements. So another important function is cell signaling and communication. Phospholipids they play very important role in intracellular signaling and communication process in cells. Signals can be transmitted across the cell membranes more easily by activating certain signaling proteins. For example, phospholipids like phosphatidylinositol 4,5 bisphosphate, that is PIP2, they are used to activate signaling pathways, including phosphoinositide signaling cascade, and this cascade controls cell division, growth, and intracellular calcium signaling. Phospholipids can also serve as energy source. So whenever the body needs, the fatty acid tails of phospholipids can be enzymatically cleaved from the glycerol backbone through a process called lipolysis. And this lipolysis releases fatty acids that can be further metabolized through beta oxidation and generates energy. So this provides energy for cellular process. And phospholipids, another important function is they act as precursors for bioactive lipids. So they, for example, they create certain bioactive lipid mediators, and this uh, to create those bioactive lipid mediators, phospholipids are essential. They act as precursors. For example, the precursor for the production of prostaglandins and leukotrienes and thromboxanes is arachidonic acid. And this arachidonic acid is released from the phospholipid like phosphatidylcholine or phosphatidylinositol. Inflammation, immunological responses, and blood coagulation are just a few of the physiological processes in which these lipid mediators play a role. Phospholipids membrane protein function. The insertion and stability of integral membrane proteins takes place in a phospholipid environment. Some phospholipids, such as phosphatidylinositol phosphates, have the ability to interact directly with the particular membrane proteins, controlling their activity, location, and function. And they are essential for cellular trafficking and membrane remodeling. That is, phospholipids are involved in cellular activities including vesicle production, exocytosis, and endocytosis. In addition to that, they participate in membrane remodeling processes like fusion and fission, which are very essential for cell division, organelle dynamics, and intracellular transport. Cellular signatures and recognition process also involves phospholipids. So phospholipids serve as biological signatures by supplying distinctive lipid profiles so that we can differentiate one cell type from the other cell type. And these lipid profiles may have an impact on processes including immunological responses, tissue formation, and cell migration by affecting the cell recognition and the cell-cell interactions. So this is all about phospholipids structure and functions. Now we will see synthesis of phospholipids. So here you are seeing choline and ethanolamine. These are both important components of phospholipids, and this choline being the precursor for phosphatidylcholine, whereas ethanolamine being the precursor for phosphatidylethanolamine. And choline and ethanolamine are major membrane lipids, and human body obtains this choline and ethanolamine through both dietary intake and endogenous synthesis, which takes place primarily in liver. So that is mostly derived from the pre-existing phospholipids. So thus phospholipid synthesis starts with choline and ethanolamine is regarded as a salvage pathway because this is going to happen from the pre-existing phospholipids. And this phospholipid synthesis takes place in endoplasmic reticulum. And here, if you see this choline is phosphorylated to phosphocholine, which then combines with the CTP to form CDP choline. Whereas ethanolamine phosphorylated to phosphoethanolamine, and then phosphoethanolamine is converted, combines with the CTP and forms a CDP ethanolamine. So here the actual how the different types of phospholipids are formed, this figure shows very clearly. So glycerol 3 phosphate is the precursor. So from the glycerol 3 phosphate is formed with the help of glycerol kinase, and then glycerol 3 phosphate forms lysophosphatidic acid. Lysophosphatidic acid forms phosphatidic acid. From the phosphatidic acid, 1, 2 diacylglycerol is formed. So this phosphatidic acid and 1,2 diacylglycerol are the precursors for phospholipids. And these are the intermediates during the synthesis of triacylglycerols. And now we have, in the previous slide, we have seen how CDP choline is formed. This CDP choline combines with 1,2 diacylglycerol and forms phosphatidylcholine, which is also called as lecithin. Whereas in the previous slide, ethanolamine gets converted to phosphoethanolamine and then combines with the CTP and forms a CDP ethanolamine. This CDP ethanolamine combines with 1,2 diacylglycerol and forms a phosphatidyl ethanolamine, which is nothing but cephalin. And then here the phosphatidic acid can be converted to CDP diacylglycerol. And then this CDP diacylglycerol combines with one more molecule of CDP diacylglycerol and CDP diacylglycerol is formed from CDP diacylglycerol. Phosphatidylglycerol is formed. So this CDP diacylglycerol forms phosphatidylglycerol. Then if you see here, inositol, CDP diacylglycerol combines with the inositol and forms phosphatidylinositol. Then this phosphatidylethanolamine can be converted to phosphatidylcholine. And here this phosphatidylethanolamine can be converted to phosphatidylserine, and which on decarboxylation, phosphatidylserine on decarboxylation gives phosphatidylethanolamine. And here one more thing, phosphatidylglycerol, which is formed, again combines with one more molecule of phosphatidylglycerol and forms cardiolipin. So this is regarding the synthesis of different phospholipids. Thank you.